Welcome to the screencast of my presentation for the IAJGS 2016 conference in Seattle. The audience was over capacity for the room, so many people who wanted to attend couldn't, and for those of you who didn't make it to the conference at all, I hope this will be a decent recreation of what we talked about on Thursday, August 11th at 9 a.m. This presentation is entitled Tip of the Iceberg because what we've been doing with Y-DNA until fairly recently has been just the tip of the iceberg. With new sequencing technology, or NGS, next generation sequencing, we can learn more about Jewish paternal lineages than we've ever been able to in the past. Using 3 Jewish clusters and 2 haplogroups as examples, I'll show what happens when we apply new techniques to old Y chromosomes. Tip of the iceberg also because we're just beginning to use these new technologies. For those of you who have been at the conference and heard Israel Pickholz a couple of times tell audiences that I've been helping him transition to his new Sephardic identity, this is just a teaser. You'll find out later on in the presentation what he's talking about. There's some technical detail in this presentation, mostly at the beginning, so bear with me. You don't need to understand every detail, and I've actually had to leave out a lot of information because this could be a 4-hour presentation instead of a 1-hour presentation. You don't need to understand biology to understand this topic. Think of it as a logic puzzle. There's a set of rules. Once you basically understand how things are supposed to work, you can figure out what you need to know and then ask questions. So first we're going to be talking about lineages. For religious purposes, Judaism is passed matrilineally, but it's extremely difficult to trace those lines because women just weren't recorded, and if they were, it was often without a surname or a patronymic. In addition, mitochondrial DNA, which is what is passed from mother to child, mutates so infrequently that a match to someone whose common female ancestor lived within the past few generations is virtually indistinguishable from a match whose common ancestor lived thousands of years ago. So, given that the female line is extremely difficult to trace both through DNA and the paper trail, we use the male line, which proves to be remarkably reliable in my experience for identifying Jewish lineages. Jewish women tend to have Jewish mates for the most part. So what do I mean by lineage? The lineage is the Y-DNA line, or what we would also maybe think of as the surname line, which is passed from father to son, more or less intact down through the generations. It can be a proxy for surname. As most of you know, the oldest recorded fixed Jewish surnames go back to about the Middle Ages, but most of the Jewish population in Europe did not have surnames, fixed surnames, until the 19th century. So when we look at the Y-DNA far back in time, it gives us some context about Jewish history and migration. As we know, a large number of the world population of Jewish people lived in Eastern Europe in the 16th through 20th centuries. With Y-DNA, sometimes we're able to go back before that and figure out where they came from to Eastern Europe. Not only that, but we can go back not just before surnames, but before the history of Judaism. If we think about the recorded history of Judaism is somewhere around 3,000 to 4,000 years, and Y-DNA can tell us before that time where people lived. So I'm going to go through a very brief primer about Y-DNA. For those of you who already know this, you might want to fast forward. The Y chromosome is passed from father to his sons, to their sons, to their sons, without recombining. That means if you're familiar with autosomal DNA and you know that chromosome 2 or chromosome 20, the 2 copies from your parents recombine and come up with a mixture of alleles, but Y-DNA, there's only 1 copy, and it's only passed from father to son. Mutations occur when a sperm cell is created, or germ cell, and they're relatively infrequently generated, but frequently enough to differentiate between branches or lineages. So the FTDNA, Family Tree DNA Company, standard tests for 3767, 111 markers. I'm not even going to talk about 12 markers really. They count repeating segments in the non-coding regions of the Y chromosome. So this is what used to be called junk DNA, or areas that are not thought to generate proteins that cause things to happen in your body. The repeating segments are called STRs, or short tandem repeats, and they're passed on to future generations, which can add their own mutations. So once a mutation happens in a father's line in an STR, then his son will typically also have that mutation, his grandson, his great-grandson. There may also be other mutations that are added over time. So here's an example of how STRs work. This is a column from a Family Tree DNA project result spreadsheet. If you see the column to the left, it says DYS 393, and there are 17 numbers below that. Those represent individuals in a project and what value they have, what number of repeats they have at the location that's known as DYS 393. So if you look down here, the repeat that happens at that particular marker is the letters AGAT over and over. AGAT, AGAT, AGAT. If you have the repetition of AGAT 12 times, then in this column you're going to have a 12. And then you'll notice that the first individuals have 12 repeats at the first 6 individuals have 12 repeats at DYS 393. The next 10 have 13 repeats. The last person has 14 repeats. And so your match list at FTDNA for your STRs will match you up with people that have the same or similar number of repeats at a substantial number of locations or markers. And I just want to mention that whenever I say the word mutation, think copy error, because a mutation is an error that happens in the copying. It's not something scary like a science fiction mutant or some horrible event. It's just a copy error. Okay, this is an example of a match list from an individual's account at FTDNA, and you'll notice over here to the left is a column that says genetic distance. So these are all the people that match this individual, and this number to the left, genetic distance, that represents how many differences there are between the person whose account this is and each one of these people at this number of markers. And I believe this is a 25 marker list. But so a genetic distance of 0, these top two, it means these two people have the exact number of repeats in each of the 25 locations that this individual has. Now, these are the Y-DNA tests that FTDNA started offering about 16 years ago. At the beginning, it was just 12 markers, then they went to 25, 37, 67, and now the maximum number of STR marker is 111. The orange button in the middle, right here, is called a tip calculator. It gives you a probabilistic estimate based on average mutation rates of how long ago you shared an ancestor with that person. So if you would click on a tip calculator for that first individual, you would get a table that would say something like you have a 60% chance of having a common ancestor with this person within the past 4 generations, for example. And it goes typically 4 generations at a time, and it'll tell you the probabilities at each of those time periods. Now, one thing you got to remember is that these are probabilities, for one, and so they're generated based on large numbers. So using a probability when you're comparing 2 people is not likely to be very accurate, because that's not what probabilities really, that's not how they work. But the other thing to remember about that is that even if you have a 90% chance of being related to somebody within a certain time period, say you have a 90% chance, you could also be in the 10% that is not related within that time period. So in order to show up on somebody's match list at Family Tree DNA, you can only vary about 10%. In other words, if you compare 12 markers, you have to match that person with a genetic distance of 0 or 1 if you're in a project with that person. At 37 markers, you have to have a genetic distance of 4 or fewer. At 67, has to be 7 or fewer. So a 60 out of 67 marker match will show up on your list, but someone who's 59 out of 67, they're one outside of that window, and that person will not show up on your match list. So that means there are a number of people that are distantly related to you, or maybe not as distantly, that are not going to show up on your match lists because they're slightly outside of that window. And that's another part of the tip of the iceberg. There are people out there that are not showing up on your match list because of the constraints of Family Tree DNA's time period window. So for a decade or so, STRs have really been the only tool available to us doing genetic genealogy. But now that we've started using new kinds of tests, next generation sequencing, which I'll be explaining in a short while, we're now finding out that STRs are kind of a blunt instrument. They don't necessarily tell the real story. For one, they're prone to back mutations, so matches might appear to have a more recent ancestor than they really do. So here's an example. Let's say your common ancestor with another individual is 6 or 7 generations back, and let's say hypothetically we know, although we can't really know, but let's say we know that that individual's value at a particular marker was 12. Let's say your third great-grandfather mutated to a 13, which is not an uncommon thing to happen. And then let's say 3 generations later, your father mutated back to 12. And then there's another distant branch, 5 or 6 generations back, that stayed at 12 the whole time. You have no way of knowing that that back mutation occurred. And maybe others that happened on both branches over the past 500, 700, 1,000 years or whatever. There could have been 2 to 3 more mutation events, but you can only see the end result, which is that you both have a 12 in that location. And that means you match, that could make you match with a genetic distance of 0. That means that even though there have been mutation events and there's several hundred years between you and that other person, the tip calculator is going to overestimate the probability that the common ancestor lived within a very recent time frame. The other thing that can happen with STRs is called convergence, which means that there can be the same STR mutation on different branches of the phylogenetic tree, which we'll talk about later, that are not coming off of the same ancestor. So this is very common in certain haplogroups, particularly R1b. If we're talking about 12 markers, 37 markers, 67 markers, there's a pretty small number of possibilities for mutations when these are haplogroups shared by millions and millions and millions of people. So it's not too surprising that 2 branches of a tree separated by thousands of years could produce the same mutation independently by coincidence. And that would make it look like a recent match coming from the same ancestor, but it's really not. It's a mutation that's been generated in 2 completely different parts of the tree, not in a recent ancestor. So this is my favorite. It's possible that you or your father or your grandfather could have had a 2-step mutation, for example, in the first 12 markers, and I'm speaking from experience. I have a family member who's in this situation. He was in the Family Tree DNA database for nearly a decade with no matches. And so at some point I finally thought, well, maybe I should check and get another member of the family to test to see if it's possible that it's just this particular individual's DNA mutations and that this is not representative of the whole family. When I did test those two other members of the family who were third cousins on different branches of the tree, they matched each other, and they also matched a whole bunch of other people, and it became clear that it was the original tester who was the anomaly. Made his mutations made him look like he didn't match anybody and like he was not related to any of those other people, when in fact he definitely was. Hierarchy, that's important. You cannot easily make a tree, a hierarchical tree, based on STR mutations because they're not really hierarchical. You can't reliably tell which mutation occurred first, which ones occurred later. There's not a clear branching pattern. If you're a project manager, you probably tried to group people based on patterns in their STRs, but if you have a large enough group, you quickly find that you see patterns which conflict with each other. And it's very difficult to find out which mutations happen in which order. And that's going to be demonstrated on the next slide. So now I'm going to show you an example in the I2A haplogroup. And this is not a typical Jewish haplogroup. In fact, at the same conference, Bennett Greenspan gave us the stats. The I haplogroup occurs in 1% of Sephardic Jews and 4% of Ashkenazi Jews. This group that I'm going to show you is larger than an STR match group. In other words, most of these people, or many of these people, don't appear on each other's match lists because the common ancestor for the entire group is farther back in time than the 24 generations allowed in a match list. And that's why projects are really useful, because it allows us to see things that are outside of just one person's matches. So in case you haven't seen a project listing before, this is what it looks like. Each one of the columns is a marker, a location, and then each row is an individual in this group. I've cut their names off for privacy reasons, but this is a small group within I2A. Now, all these numbers here, these are just the numbers of repeats for the different markers. If you look here, this is one column, this is the first column, DYS 393. Everybody in this group has 13 repeats at this location. The pink and purple squares, those indicate places where that individual's allele or value is different from the mode or the most common value for this little group. The pink ones mean their value, that individual's value is higher than the mode, and the purple means they are lower than the mode. Now, if you notice, this spreadsheet is actually sorted. The FTDNA does kind of a very crude sort. They start with just the first column, sort on the first column, then the second column, then third, then fourth, and so on. This doesn't necessarily mean that this is a good way to group people. As you'll see in a second, your eye will start to pick out patterns in these sorts. So you notice here's a group of people who have 17 there, at that particular location. The most common is 16, and there's a few people with 15, but this group of 5 has a 17 there. So that looks interesting. I wonder if that means that they are a closely related group compared to the rest. Then you notice down here you have this 24. These people, maybe they're more closely related. That looks interesting. And as I said, your eye can start picking out patterns. Then you notice, oh, look here. The mode here is 12. There's one person with 11, but we have 3 people together with a 13. I wonder if that means something. And then we notice over here, a group of people have 22 here instead of 21. And then we notice, the same people that have, 2 of the same people that have a 13 also have a 22. That's interesting. Oh, but then this guy down here has a 22 also. And they have different values for these other markers. So where you start to see patterns, they're not necessarily indicative of a close relationship. Here's another little pattern. And you notice, well, this might mean something. People have a 13 and a 22 here, also have a 12 here, these two guys. However, they have a different value at this particular marker. So you can see how it would be very difficult to take all these different numbers and try to sort them in some way that would show you an evolving set of patterns. Then, to make it more complicated, so these people outlined in blue, these are all the people that have known Ashkenazi or Sephardic Jewish ancestry. So we have one person up here, one down here, these several in a row, and then this person down at the bottom does not. So what do we make of all these people who have a common ancestor? We don't know how far back it is, and we can't really tell when those different lines diverge from each other. So what do we do next? We have something called next generation sequencing. So we use, in next generation sequencing, we look at something called SNPs, single nucleotide polymorphisms, instead of SGRs. And what this means is, at any given location on the Y chromosome, you have one of 4 nucleotides, A, C, G, or T, and you go to look at that location, or the sequencing machine goes to look at that location to see what letter do you have there. Do you have an A, a C, a T, or a G? If you have a G and the reference value is T, then that is a mutation. That's a change. It's a SNP. So a SNP just means a change in one letter, one nucleotide, somewhere on that chromosome. That applies to all chromosomes, not just the Y chromosome, but we're only talking about the Y right now. So SNPs have been around for a long time. We've known about them for a long time. That's how the original haplogroups, the A through T haplogroups, were created, by looking at where large divisions, large branching areas in the haplotree occurred, and what SNP or change those people have in common when they split off. In the past 3 years, though, that technology has become inexpensive enough to test large numbers of people at the level of 10 to 12 million locations on the Y chromosome. In 2013, FTDNA introduced the Big Y test at the 2013 Administrator Conference. But they had a project before that called Walk the Y, which had sequenced a number of Y chromosomes to try to see if they could map out a structure. The test scans millions of locations looking for those changes, and that allows us to create hierarchy and layering when you pull together many, many tests and you start to look at which SNPs do people have in common, which ones do they not have in common. So when you come to a branching point, the question is, do you have this mutation, yes or no? If it's yes, you go down one branch of the tree. If it's no, you go in a different branch. Those are mutually exclusive, and it makes it very simple to see how those SNPs evolved. You can't be positive for 2 SNPs that take separate branches down the tree. If you are positive for 2 separate branches, then and it's a reliable SNP, then the tree has to be wrong. And what happens is that mutations can be a proxy for the passage of time. And we have a number of different methodologies for trying to estimate how long the time is between generation of different SNPs, which is at this time just estimates, and we'll talk more about that later. So first, I'm also going to talk about a couple of definitions. There's something called the reference genome, which is also known as the reference assembly. It's a DNA sequence database assembled by scientists as a representative example of a species. For example, the Genome Reference Consortium Human Genome Build 37 is derived from 13 anonymous volunteers from Buffalo, New York. In other words, it's a standard model to compare people against. If the reference genome has an A at location 1, 2, 3, 4, 5, 6, 7, 8, and you have a G there instead, you have a SNP, or a mutation. So in order to create a phylogenetic tree, we map all the mutations that are found on all the Y chromosomes, then figure out which SNPs are shared and which ones are different. So you've probably seen some version of this kind of a chart before. It's showing the entire phylogenetic tree for the Y chromosome in humans. And it used to start at A, but it now starts at A00 because they've found a branch before A. But you'll notice here that at each branching point, there's a combination of letters and numbers, and that represents a SNP. So millions and billions of people all start at this very beginning. The people who have this mutation or SNP at this branching point move down here. If you're at this point, if you have mutation M60, you go this direction. If you have the mutation called M168, you go that direction. If you're on this branch and you have either M174 or M96, you go this direction. If you have P143, you go this direction. And all of these SNPs define these large, what are called haplogroups, clades, of the human Y chromosome. And basically what happens is as you move farther and farther down the tree, there are fewer and fewer people that you share lots of common SNPs with. So you go from SNPs that you share with millions of people, let's say R, the R subclade here. This M45 is shared with millions and millions of people. The farther you go down the tree, the fewer people you share SNPs with. You get to the SNPs which you share with smaller numbers of people, and then which SNPs you share with very few people, and then the ones that you share with nobody. That's how the hierarchy starts to emerge. This is the most bird's eye view of the kind of phylogenetic tree. I'll show you some examples of some more specific ones in a couple of minutes. Okay, the kinds of tests that we use to find SNPs. First of all, SNP data for you or someone in your family is completely useless without someone to compare it to. In isolation, it's just a long string of letters and it doesn't really tell us anything. So you need to combine your data with a lot of other people so that you can start to see the patterns. So Big Y and Full Genomes Elite, which is a competitor to Big Y, those tests scan 10 to 12 million or more locations on the Y chromosome, and then they compare at each one of those locations, they compare to the reference to discover all the shared and private SNPs. This starts to create the skeleton of the tree. So giant databases like catalogs of known mutations in the human Y chromosome begin to be created. One thing to know is there's no one catalog or one reference that has all of the mutations, all of the SNPs. They're scattered around in several different databases, and I'll talk about that later. These tests, Big Y and Full Genomes, can cost between $500 and $800, depending on if there are coupons or sales or what's going on at the time. Then we have SNP packs from FTDNA or a company called Y-chromosome, which has SNP panels. What this is taking the strategic branching SNPs that have been discovered in other people's Big Y and Full Genomes Elite tests and putting them all into a hierarchical group of SNPs and testing just those strategic SNPs. It doesn't tell you any SNPs that you have that are unique to you. It just tells you yes or no, do you have the SNPs that have already been discovered in other people and put on this particular panel. And they usually contain 100 SNPs or so, and they cost between usually $80 to $120, $130, depending on which pack it is. And usually these are by haplogroups, so there will be a SNP pack, for example, at Family Tree DNA, if you're R1b M269, M269, you can get a SNP pack that will just go down the upper branches of R1b or RM269 and test you against each upper branch, and then go down some of the side branches to give you a main branch, but it won't discover everything that you have in your chromosome. And then once you have a group that has its tree fairly well mapped out, and you know which SNPs people share in common, which SNPs seems to be private SNPs, you can then test some SNPs with Family Tree DNA, but their catalog is somewhat limited of the SNPs that they will test individually. And then there's, again, Y-seq will make custom SNP tests for you, and sometimes custom panels. And those range from $17.50 to about $49, $50, something like that. But you have to have a group with enough big Y or full genome results to know which SNPs make sense to test against which people. We're going to talk about variants, which is another name for SNPs. Every test result variant has a position, a reference value, and the value in that particular sample. So a position is usually an up to 8-digit number, which is a place on the Y chromosome. A reference value meaning, is it an A, C, G, or T that's in that reference sample? And then the value in your particular sample, is it an A, C, T, or G? And there are several ways that this can be expressed in the results. Reference is, again, that's a sort of combined a combined genome composed of several different people's values, versus derived, and that means you. So reference versus derived means the reference versus your result. Ancestral versus derived, it's the exact same thing, it's just different wording. And then there's reference versus genotype. That means the same thing. Genotype means your particular value at that location. So SNPs that are shared by more than just an immediate family get named. If you and your brother share a SNP, that's not of much interest to the genealogical population, so that's really not going to get named. But if you have a SNP that seems to be from an ancestor 600-700 years ago, and particularly from people with different surnames, then generally that SNP will get a name. Now, the tricky thing is that there are different labs and companies and studies and different people that have given out names to SNPs starting several years ago. Fortunately, the ISOG website, the International Society of Genetic Genealogy, they have a list of all the prefixes, so that if you see a SNP that's named a particular way, it's usually a letter with some numbers, you know who named that SNP, and you can also kind of have a sense of roughly when that SNP was discovered. All these different organizations that are naming SNPs, people in general try to use the name if there's already a name, but it's often the case that SNPs get discovered in parallel, or they get discovered by someone who doesn't have access to some SNP catalogs of things that were named previously, so they'll give it another name. So sometimes some SNPs have more than one name, and it doesn't mean anything. It's just a label. It doesn't have any value. It's just that you need to know that it's the exact same SNP. There is no central repository database or catalog of all the name variants and all their multiple names, although in various places there are near complete collections, and sometimes you have to check multiple places to figure out what the location of that SNP is and whether it has multiple names. And the databases I use the most for this purpose are Y-full and Y-browse, and that'll be on the handout that goes along with this presentation. Now we're going to get into some specific examples. So I'm going to give you a couple of examples from the subclade called I2A, haplogroup I2A. And this is a map from Wikipedia, which shows the present-day distribution of I2A across Europe. It's most prevalent in Slavic areas, and you can see by the darker colors that it's most prevalent there and in Sardinia. And also there are some pretty big clusters in some areas in Iberia and some in the UK. But remember that this is present day distribution of people in the I2A haplogroup. This may or may not mean anything about ancient DNA. Until we have more ancient DNA samples, we don't really know how close present day distributions are to ancient ones, but we can assume probably not. This is probably not an accurate map of the ancient world. If you look at the Middle East, you can see this is not particularly a Middle Eastern haplogroup, at least not around Israel. And as I mentioned before, it's about 4% of, well, actually, all of I is about 4% of Ashkenazi population. Now, where do they get these distributions? Well, they're basing this on where people who have tested where their ancestors were in the last few hundred years. So we don't really know if someone's ancestors were in these locations 200 years ago. We don't really know where they were 500 years ago, 1,000 years ago, 2,000 years ago. So this is always the problem with extrapolating into the past from the present. Until we have lots of samples of ancient DNA, we're really just guessing. So here's a tree that I made from the project spreadsheet that I showed you before that had the various different patterns that we couldn't really tell which branch came from where, and it was just there were just various different common SNPs that people had, and I'm sorry, there were different. This is based on the STR spreadsheet that I showed you before of that cluster within I2A. So think back to the STR values for the group in the spreadsheet I showed earlier, and remember how difficult it was to make sense of the groupings. There are some ways that we can try to make sense of it using some online tools and some other free tools that try to create phylogenetic trees out of STR data. So I used a website called McGee's Online STR Calculator, and which generates the data that goes into a program called Philip, which has many different algorithms that try to take this STR data and fit them into a reasonable tree based on how close they are on various different markers. This is just an approximation, and they really are just trying to fit the data into something that kind of shows branching. This one uses different statistical methods, and that's all just contained in the Philip program. I use an algorithm called Kitch, but there are lots of different algorithms that are within the Philip program. And then I use a program called FigTree to actually generate the tree. So starting from the right, these over here, these are individuals that were on that spreadsheet, and these are only the people that tested 37 markers or more. And I only looked at the first 37 markers so that I was comparing apples to apples. And you will see here that we have someone from Italy, someone from Portugal, somebody who doesn't know where his ancestors are from, Switzerland, France. This right here is what I call Jewish cluster 1, because all of these individuals in here are have known Jewish ancestry. Then we have an individual from Iberia, one from Italy, and then we have a second Jewish cluster down here. So the way to look at this, the way to read this this phylogenetic tree is you're starting here. As you go to the left, you're going back in time. When you come to a node, that represents the common ancestor for the people that are connected to that node. So this is a common ancestor for these two individuals. If we go back here, this right here is a common ancestor for these two plus these two. We go back another level. This is the common ancestor for all of the people over here and this person, and so on. The fact that we have two different Jewish clusters made me think that possibly the common ancestor for the Jewish individuals would be back here, because this is the first node that encompasses both of these two clusters. If that were the case, then that would indicate that this person here from Iberia and also this person from Italy would belong within this Jewish cluster, and also these people from France. But the important thing to note here, all the people that are in green have been identified as having this SNP PF6950 by the test that they did, which was Geno 2.0. So all the greens are Geno 2.0 or someone who tested the PF6950 SNP individually. And all the blues are people that we did big Y. So we already had a couple of big Ys available here, I believe. This one. And then what we tried to do is get a big Y, two big Ys within this larger Jewish cluster, and then one from down here, so that we could begin to see the structure. So we try to get these big Ys spread out along this tree. And now, so remember, you're moving back in time as you go up this tree, and you get to this PF6950, which is a SNP that all of these people share, but probably there are other SNPs that are going to divide up these branches. But we're assuming there's two Jewish clusters, that there's this Iberian person and this Italian person are probably somehow within this Jewish cluster. So, in fact, when we got the big Y results back, we found out something very different. In fact, the Jewish cluster here, all three of the Ashkenazi individuals that did the big Y are all within the Jewish cluster. And this Italian person over here, person of Italian descent, is over here, in fact, and not within the Jewish cluster. So let's see how this works. So we're starting up here at P6950, which we know they all share, and there's an additional 14 to 17 SNPs that they all share. But then here's a branching point where everybody below here has a SNP called PH1671 that these individuals do not have. And all of these individuals have 21 SNPs in common with each other that this branch doesn't have. So you can see that we thought these were going to be two much more separate clusters, and this person was going to be somehow over here. And in fact, when we see the SNPs, it didn't work out that way at all. And then, so we noticed that these two people that were in cluster number one definitely are more closely related than they are with the third person. We think their most recent common ancestor could have been sometime around 1200 AD or CE, and that their common ancestor, all three of them, is more like around the year 700, something along those lines. Their common ancestor, the common ancestor between these two branches, is probably around 3,000 years ago, around 1000 BCE. So this Italian person and these people, their common ancestors about 3,000 years ago, which is not what it looked like at all from the STR tree that we made. For comparison purposes, I've also included, here's an individual that was in a study that was done in 2014, and this person who had this SNP, these two SNPs shared with this Italian person. This person's from Greece. So the other thing to note is this Jewish cluster has about 21 SNPs in this box here that these individuals over here do not have. This is a really long time to not have any branches. So normally each time there would be a SNP here, that's a branching point. There would normally be lots of side branches for these 21 SNPs. And so that could be indicative of a bottleneck, which is a long process of mutation where normally there would be multiple parallel branches at each mutation, but the other branches either did not survive or have not been tested. And when I say did not survive, I mean there are known bottlenecks in Jewish history, places where large numbers of people were killed, and there's a small number of founding ancestors that then sort of restart the population. So that would be examples like the Black Plague, that would be the Crusades, various pogroms in Europe, and so on. And of course, also the Holocaust. And this is very unusual in a project to see this big of a cluster. When I work with other haplogroup administrators in the haplogroups above me, they're always very surprised when they see these large blocks of SNPs, because this is not what this kind of a tree would typically look like. And I want to emphasize that it's really important to work closely with the haplogroup administrators upstream from where you are, because this is how we get migration context for the Jewish clusters. And I've had very good success working with, for example, the I2A administrators. I had a family member that was in one of these clusters, and so I contacted the I2A project administrator and I said, I see some Jewish clusters in here. I'd be very interested in working on these and trying to break them down further and see what I can learn about them. And so, how do you feel about me working with you as a co-administrator and working on these clusters? And they said, great, the more the merrier. And they welcomed me into their group, and now I'm working on some of these clusters in I2A. I want to make note of the dates that are in here, 1000 BCE, 500 BCE, and so on. We don't really know exact dates. These are dates that are derived by looking at how many SNPs are not shared, how many SNPs are shared, and the guys at YFUL have digested lots and lots of data and written a white paper and said, here's our method for calculating the average number of years between SNP mutations. It's not exact, but it's not likely to be orders of magnitude different from their estimates. But just remember, all of these studies are still in their infancy, and we don't know exactly how many years each one of these branches split off, but we have estimates, and we're doing our best to continue to refine those with more data. So summing up, a tree based on SNPs like we have here looks really different from the tree that's based on STRs. So now we're going to look at a different cluster within I2A, and this cluster is very remote from the cluster we just looked at. They're separated by several thousand years. This particular cluster is known as the Dinaric subclade for the Dinaric Alps, which is a mountain chain across Albania, Bosnia and Herzegovina, Croatia, Italy, Kosovo, Montenegro, Serbia, and Slovenia. Base this chart just on the STRs of the Jewish cluster within this particular subclade, and I'll show you how they are situated with the non-Jewish part of it in a minute. But this is a much bigger cluster, as you can see. This is based on 37 markers, and Same thing. If we look here at, so we had one big Y already here, and so we had an idea of what the subclade was, and this is actually called A2512. And we then also had another big Y pending here, and we wanted to try to find at least one more big Y to do somewhere outside of this area here, because if you look at these two people, their common ancestor looks like it meets up around here, so we don't really need another person from within this whole group. We would really like to have someone outside of that, so even farther back, like someone here, down here, or what we finally ended up with was somebody on this branch right here. And eventually I would like to get some more data from these sides down here, because as you can see, they have they appear to be farther away based on their STRs. But we ended up with this person who meets up with this other cluster all the way back here. So that seemed like a reasonable group of people to test. And what we would expect to see is that these two would be closely related, and then this person would be outside of their cluster, that there would be some mutations up here and up here that would separate them into separate branches. So once we've done a big Y, we get a completely different story again. We have, here's A2512, which was the SNP we already knew about before we did the big Y tests. We found another SNP called A10959 that separates the Jewish cluster from this other cluster here, which I'll talk about in a minute. And within this Jewish cluster, we have two branches. This is the person, one of the people that look close together. And in fact, what we find out is these are the two people we expected to be closer together, and this person to be the outlier. But in fact, when we look at all their SNPs and we see which SNPs do they share, which SNPs do they not share, we find that these two people share at least 3 SNPs. We think it's actually 4 now, that put them fairly recently related to each, with a common ancestor who's fairly recent. And then the third person is fairly far back, somewhere, actually, that's not correct. So this is actually a couple of hundred years earlier. But what's also very interesting about this is we have our Jewish cluster here. If we go back to this SNP, which everybody below here has, which looks to be around the year 0, what we have over here is an individual from Greece. We assume non-Jewish, we don't really know. But here's a person whose ancestors were from Greece on this branch with the Jewish cluster, with a common ancestor around the year 0, around the Roman times. And then when we go up and over here, back to around 200 BCE roughly, there's another branch over here and another person whose ancestry is from Greece. But in fact, this person from Greece has done a big Y, but there's actually a whole cluster of a bunch of other people from Greece who have not done big Y, but who appear to also be on that branch. And there's only one person on that branch that's not from Greece, and he is an individual from Mexico, but his surname is Griego, which in Spanish means Greek. So the other thing to note is there's kind of a consistency when you look at these private SNPs. This means SNPs that are not shared by anybody else, and you add them up. This person is about 16 SNPs back to here. This person is 13, 14, adding those together. We have 5 and 10, 11 back to here. These numbers are reasonably consistent, which means this tree structure makes sense. Now, this is a haplotree of the entire I2A haplotree. This up here is the first, where the first group I showed you is located, the PF6950 people. And this is the second cluster I showed you just now, the A2512 people in the Dinaric cluster. This first group is in what's known as the Sardinian cluster, which is most common in Sardinia, but also a lot of Iberians, also some other Mediterraneans, and this Dinaric cluster we've seen breaks down into further into another group that seems to look kind of Greek. Now, this I2A haplogroup has about 3,000 members in it. And this is one of the haplogroups. This is a haplogroup that Bennett Greenspan likes to point out that is present in only about 4% of Ashkenazi DNA, and it's also known to be a Slavic, more common in Slavic areas. So he likes to refer to this as, you know, a Jewish haplogroup that is due to the Cossacks coming through and raping people. And I've not studied all the Jewish clusters in the I haplogroup, but at least in these two clusters, the closest common ancestor with any Slavic branch of I2A is in the range of like 2,500 years ago. So it's long before any Cossacks. It really appears to me that both of these clusters, their common ancestor a couple of thousand years ago with the non-Jewish branches is probably from Greece or the Mediterranean. People are going back and forth across the Mediterranean all the time in the ancient world. You've got a Greek or a Hellenic dynasty in Egypt in 300 BCE. It just takes one guy to start a Y-DNA lineage, so I really think it makes a lot more sense to postulate that these two clusters are Mediterranean, Greek, Italian, you know, we don't really know, but they're at least 2,500. The closest Slavic common ancestor is probably 2,500 years ago. And with this cluster in particular, it's probably even farther back. The very beginning of I2A, which is also called IP37, is some 20,000 years ago. I don't think they have much to do with the Slavs. Okay, now I'm going to talk about a little bit about the technical stuff about how do we make these charts, how do we figure out how this branching happened, and then after that I'm going to talk about another sample cluster that I'm very excited about. So here's a quick note first about Big Y's matching function on their website. When you go to look at your Big Y, if you've done a Big Y and you go to look at the Big Y matching, you're going to be very confused, and I recommend to people don't even look at it because it doesn't actually work very well to give you an accurate picture of who your close matches are. I just ignore the order of the list because it purports to show you at the top of the list the person you're most closely related to based on the number of SNPs you share, which is not a good way to measure, and I'm going to show you in a second what that looks like. So the trick to see the matches that are actually closest to you is to use the pull-down menu for shared novel variants and go to the very bottom of the list, the SNPs you share with the smallest number of people. So here's a screenshot of Big Y results, and you'll notice here this is just a portion of that screen. Shared novel variants, I've used this pull down. Instead of saying all variants, I've gone to very, very low to the bottom of the list, and I've found a particular position, a variant, a SNP, that is shared with only 6 individuals. And when I take a look at who shares this SNP, and I look at the list. These are in fact a cluster of people that, based on the SNPs, that this person is more closely connected to. So if you pull this down and look at the lowest, smallest number of group of people that you share novel variants with, those are going to be your closest matches, not the people at the top of your match list. All right, when you do a big Y, there are 3 data files that you get. One is a CSV file, which is the digested spreadsheet. You can pull it up in Excel or any similar program to that. It's only showing you high quality variants. So in other words, when the machinery in the lab has read all the slivers of DNA, it looks at how many times has each area been read, how satisfied, how high is the quality of those reads, and it throws out, the CSV file throws out anything that it deems inaccurate. So that makes it a very conservative list, and it's probably leaving out some that are relevant, but that's the easiest file to look at. There's another set of files called the VCF embed files. It's a zipped, those are zipped together, and it's 2 spreadsheets that are more extensive. The VCF file has all the SNPs, even that weren't read many times, and it'll give you indications of how high the quality is for that read. And then there's a embed file which tells you what the coverage was. So if there's a particular SNP that you think a person should have been positive for, but in the VCF it's not showing that they're positive for that SNP, you can look in the embed file and see, was that particular region where that SNP is located, was that covered in this particular big Y test or not? And if it wasn't covered, then you don't know if it's actually negative for that SNP or just missing. And that's an important thing to know, is that big Y files are not necessarily consistent. One person may have 100 reads at a particular location. Another person at that same location might only have 3 reads or 0 reads, or what's called a no call. They're chopping up the DNA into small segments and reading it multiple times, and it's not necessarily consistent from person to person. So that's why we need this other file called the BAM, which is the biggest raw data file. It has all the information about how many reads at each location, what was found there, was it a bunch of T's or half T's and half G's, or just shows you all the raw data, and you have to have a special program to actually read that raw data. That's also the file that you will send or upload to the Y-Full company if you choose to have them do an analysis of your data and put you on their haplotree, for which they will charge you a fee. Same for Full Genomes Corporation. They will need that BAM file if you want to have an outside service to interpret the data for you, which I do recommend unless you become very familiar yourself with analyzing these data files. The BAM file is about a half a gigabyte, so typically FTDNA will give you a link to that file, which you can then give to someone to download rather than trying to email that file to someone who's going to analyze your data for you. So this is an example of a spreadsheet that I use. When I first get a result back, I use the CSV file and plug it into a spreadsheet that I've already created. This is an example from a subclade of J2, which is the BackRack project. This is actually just a tiny section of the spreadsheet, but I want to give you a sense of what I do with the data when I see it. I sort that person's data by position, which is this column right here, and then derived, which is a true or false, yes or no field in that CSV file, and then SNP name. But mainly I'm looking at this level, I'm looking at positions, I'm trying to find places where groups of people have positive SNPs, have the SNPs in the same location. These yellow and blue areas are upstream of the area that I'm looking at, which means it is farther up the phylogenetic tree than this particular group. So just as an example, this individual here at position 1503424, the reference value here is a T. This person has a C. All the rest of these people have the reference value, which is a T. So I've left those blank, but this person has a C. Same person at 155506284, where the reference is A. This person derived, meaning their change is they have a C there instead. And so I put those in pink so I can easily see them. The interesting part here would be the mint green. So the mint green, these are at the level of this particular project. This here is Y11782, which is one of the defining SNPs for the pre-Bacharach branch of the J2 tree. And as you see, everybody in this group is positive for this SNP. And they're also all positive for the SNP, and that's at that level. Now, you notice the dark green here, there's Y13373, also known as FGC 20419. At this position, the reference is an A, and the mutation is a G. All these people have it, but this person does not. So he is in the pre-Bacharach group rather than the Bacharachs, because the only people who are positive for this either have the surname Bacharach or have STRs that are extremely close to the people with the Bacharach surname. And then other interesting thing, if you see Y13097, this is shared only by these two people, and so is this one right here. So they form a distinct branch from everybody underneath this dark green. So I always make a first pass doing this by hand because I'm very familiar with the SNPs here. There is a guy named David Carlisle who wrote a program some time ago that will compare groups of VCF files, and it works reasonably well, and it gives you a table, a spreadsheet that's pretty accurate, but I actually like to use my own color coding system, so I do this part by hand. This is a program called IGV, Integrated Genome Viewer. It's free. It's easy to use once you learn what you're doing, but it's not easy to install unless you're fairly familiar with at least on the Macintosh, you have to be fairly familiar with compiling programs and so on. So if you want to use this on your own computer, you might need expert help. So this is Y11782, which I showed you on the spreadsheet. It's at position 16172,650 on the Y chromosome. So I just plugged in, actually I put that number 16172,650 dash and then the same number, but it shows you a little more around the periphery of this particular location. And you can see here the reference value down at the bottom is a G, and these three individuals have A's here instead of a G. And if you look at this green color block, you can hover over that color block. It will tell you how many reads that person had at that location, and it'll tell you were they all, if there's 100 reads, were they all A's, were they partially A's, partially some other letter. Occasionally you'll see something that's partially the reference value and partially the mutation value, and those are gray areas, which I'm not going to get into here, but anyway, these three are all positive for that Y11782, which is the SNP that defines the proto-backrack, pre-backrack branch. All right, talking about testing strategies within a group. If you have a pretty large group, I highly recommend to work with the haplogroup administrators for the main group or subclade. So if, for example, those I2A groups, I work with the haplogroup administrators there to ask their help in first recruiting people into the project, but also they help with analyzing data and so on. And then in all these groups, we go through the match lists of all the people that are in the project, and we invite those people in so that we can analyze their information against the other people. And then I also go through other projects besides the haplogroup projects, like for example, in this group, I would go through the Greece DNA project, some other projects that might have other people who might be within some of these subclades, but they're not members of the I2A haplogroup project. And then I also use the genetic distance matrices to plot individuals who are farthest apart in using the McGee online program. Because again, we don't want to do big Y tests on people that are very, very close to each other. We need to spread them out a bit so that we can start to determine the boundaries of that particular cluster. And then so we don't want people that are too close together, but we don't want only people who are extremely far apart, because then we don't know when someone has, say, 10 private SNPs, we don't know if those are actually private SNPs, or if that's indicative of another branch where the people on the other side don't have those SNPs because it's a different branch and not because it's that person's private SNPs. And two other terms for private SNPs are singletons and novel variants. And then what I typically will do is try to create a STR tree as best I can, and then create a tree based on the SNPs from the big Ys. So we have a scattering of big Ys, and then we create a tree, and then we look at people's STRs and see who are they most similar to, and we test either SNP packs or individual SNPs of those people, so that we don't, if we have 50 people, we don't do 50 big Ys. We do maybe 10 big Ys, and then we try to guess which SNP branches the other people who didn't test big Y might belong in, and then test them just for those particular SNPs. So here's my last example, and this is really my favorite example, and one that's been taking a lot of my time over the past 10 months or so. This started out with a group of 4 people who were STR matches with each other, although not close STR matches, but they had independently done big Y. And we had submitted their VCF files to a guy named Alex Williamson, who is a volunteer who has created something called the Big Tree, which is for everyone who is on the R1B branch called P312 or below P312, which is a lot of people, including his own Scottish clusters, and so that's why he became interested in this particular area. As far as I know, he's the only person that's doing a project like this, but we were lucky that we fell into this particular project. So they ended up on this tree, these outlined in blue here. These are the 4 Ashkenazi guys who had tested, and here's what Alex came up with in his big tree. So I first took a look at this, and I noticed next door, here's this other cluster. with a common ancestor, I couldn't really tell how far back that common ancestor is, but I look and I see the first name on that branch is Simois. So that's a person named Simois, his last name, and he's from Portugal. And I looked at some references and found that the name Simois is actually the Portuguese version of the first name Simon. So I thought, that's kind of an interesting first name. Doesn't seem real typical of a Portuguese surname, but of course I didn't really know a lot about Portuguese surnames. So I contacted him and I said, is there any chance, or do you know of any possibility that you may have had Jewish ancestry at one time, maybe Converso family? And he responded and said, I don't know, it's quite possible. I don't really know anything about my father's family, and in fact they are from the northern corner of Portugal, which is where a lot of the Conversos hid out around the Inquisition time. And so I was in touch with another person who had originally done the Big Y test, and we decided to start a project. And we initially had thought we would start a project focusing just on these two clusters here, this branch right here. And we talked to the DF27 administrator, who is basically the next major project above us, and he felt, this is going to be too small of a group. What if you go up a couple of levels and focus on this SNP right here? And we looked over here and thought, okay, well, that's interesting. There's a lot of Hispanic people and a couple of other, this is a person from Ireland, a person from the Netherlands. And so we said, okay, fine, we'll focus on this SNP FGC 20747. And by the way, this particular SNP was discovered in the Big Y file of this person when it was analyzed at Full Genomes Corporation. And so it was named. And then once it was named, all these people that were positive for this same SNP, it had a name. So this is the Upedia map of DF27, which is the main branch above us, and it's most common in Spain and Portugal. But again, remember, this present-day distributions are not necessarily indicative of ancient distributions. We don't really know when and where this particular DF27 SNP happened. There's a lot of discussion about that on various message boards, but it's commonly found in present-day Iberia. We started inviting people from the STR match lists and then the matches of those matches, then other people from the DF27 project who had done SNP packs, or people whose STRs look similar. And eventually, after many months of doing Big Ys and individual SNP testing, we end up with a tree that looks like this. So we have more than 150 people in the project at this point. So the people down here at the bottom with the blue stars, these are all people that have Jewish ancestry and, at present day know that their ancestors were Jewish, mostly from Eastern Europe or Germany, Netherlands. And then we have one person who knows his ancestry is Sephardic and whose ancestors were more recently in Turkey. So given that all of those people at the bottom are definitely Jewish, then we can infer that the next ancestor above them, the place where those branch, that person was most likely Jewish also, because to have multiple Jewish descendants, probably those branches were also Jewish. And if we go up here, we can infer that that ancestor was likely also Jewish, because everybody below him is present day. And then same thing over here, we can infer that these branches are all Jewish and that one as well. So if all three of those branches are Jewish, then we can infer that this ancestor here about 1,000 years ago definitely was Jewish. We don't know about the ones farther back, so that's a big question. What about the people farther back? Well, we have this SNP here, farther back, which is shared with the Jewish cluster, and also these two guys, one from Brazil and one from Portugal. We don't know if they had Jewish ancestry. Based on their trees, I think it's quite possible. If we go up another level, so that we're now around 2,500 years ago, and now we have a shared ancestor with all the Jewish members of the group, and then also this Brazil and Portugal, and also this person from Brazil, and also a person whose oldest known ancestor was from the Philippines. Again, that's an Iberian territory that was discovered by the Portuguese were there before the Spanish, then the Spanish claimed that as a territory of theirs. If we go back even further, then we have a common ancestor over here with more Portuguese, someone whose ancestry we don't really know. Going back further, we've got Spain, Portugal, and then all the way back with a common ancestor. This is estimated about 4,000 to 4,500 years ago, based on Y-Full's estimates. I tend to think it might not be quite that long ago. I think it might be more in the range of 3,000 to 3,500 years, and here's the reason that I think it may not be as long ago as that. Sorry, that's the Jewish ancestor there. Because down here we have a man from Belarus who also descends from a Sephardic family. So if his common ancestor with the rest of the Jewish cluster over there is all the way back here, then that brings up questions of whether that's a coincidence or whether that means that the Jewish ancestry goes back further. Many of these people down here have family trees that look like they could be potential conversa. We have not been able to prove that yet. We also have one person, this person down here, whose oldest known ancestor is from England, but when I talked to him and said, looks like, this looks more Portuguese than English, and he said, yes, we think, we have reason to believe that the oldest known ancestor here from England was actually a Portuguese Jew who settled in Virginia in the 1600s under an English name. So we have not tracked that down yet either. But if we have Jewish descendants all along here, then the question is that a coincidence, or do we have a Jewish ancestor much further back than this 1,000 year ago Jewish ancestor? We don't know. These are things that we're still studying, and we're going to continue working on that and investigating. So that Jewish cluster that was within the largest group, we've done a fair amount of SNP testing below that SNP where we know the Jewish ancestor was, to try to see if we could group them geographically, to see if we could figure out how recently they were all related to each other. And as you can see, we've got some of these down within the last 500 years, 400 to 500 years. And then over here we have those individuals that are slightly farther back, but still vaguely within this area. The blue are people who have done Big Y, and the pink are people that we've been able to test just individual SNPs based on how close we think they are to the people who have done Big Y. And we've been able to sort everybody fairly finely within this cluster because there are so many of them, for one. And then now I'm going to switch to a Google map to show you the geographic clustering within this group. So going with the smaller clusters that we've been able to find using SNP testing, I'm going to show a little bit about the geography of how these SNP groups break down. So here's one group that's relatively closely related within the last 500 years, and you can see the pink marks there, and those are two in Germany and one in the Netherlands. That's a fairly close together cluster in terms of the grand scheme of Jewish migration. And then the next closest cluster to those people are these three over here. And in fact, we have reason to believe that this mark is going to move more down this direction when we get more information. Then we have another small cluster, these yellow ones. These all share SNP within about the last 500 years. And then we have this cluster, these two green ones. They share an ancestor within the last 450 years. And then the bigger group of which they're a part, more like within the last 600 years. And you can see the geography there. Then another subcluster here within the last 600 years. Those look fairly close together, except for that person in Western Germany, which is kind of an anomaly, and we don't really understand why that is, but it's definitely an outlier. And we don't have all the geographic data for all the members of the project yet, so we'll be continuing to work on that and see if these maps still make sense when we have everyone in there. This is a cluster that's much further back, 800 years ago, so you can see they're much more spread apart, but except for the person in Belarus, they're more toward southern, southeastern Europe as opposed to northeastern. And then we have just one person who is from Turkey coming down there. So if we look at, if we put everyone on the map all at one time, here's what it looks like. And this part is a work in progress, but we just thought it would be very interesting to see if we could notice any geographic clusters. So going back to Alex Williamson's big tree from about a year ago, you can see there's the original 4 testers and the side group of the potential Portuguese, maybe Converso person. And now here's what it looks like. And here are the 4 original testers and the 2 others in that side branch. And you can see we've expanded this by a tremendous amount. And we actually have new results that still need to be added. And finally, this is all done by citizen scientists, not just me, but the administrators of the other haplogroup projects and people like Alex Williamson, and lots of other people who have spent lots of hours analyzing all this information and trying to understand it and teaching other people and answering questions. For the most part, we're not biologists or geneticists. There's no textbook for what we do. A lot of people ask me, where can I read a book about how to understand these results? And there really isn't one. We just have to get our hands dirty. We roll up our sleeves and we ask a lot of questions on forums. Most of the haplogroup projects have message forums that you can join when you join their haplogroup project. There's really no substitute for getting your hands dirty. I can understand these things in the abstract from a distance, but it's not until I really started making spreadsheets and looking at the data and looking at lots of data that I really can understand how this all works. And I can't, again, enough emphasize how much expertise there is out there that people gladly share. And that's it. I have a handout, and here's the URL of the place where the handout is downloadable, and thanks a lot for your attention.